Hi, everybody. In today's video, we're going to go over the origin of eukaryotic cells, the kinds of cells that we're composed of, as well as fungi and plants. Now, I'm going to highlight three differences between the bacterial cells that we talked about last time and our ancestral cells, which were archaea. Now, I'm going to call this a proto-eukaryotic cell, and you can see this on the right, is first much larger than the bacterial cells we talked about last time. Now, this is not perfectly to scale, obviously. I'm exaggerating here, but these cells were much larger. The second major difference between our ancestral cells and bacteria were that our chromosomes, instead of being single and circular, were actually multiple and linear. So you might be aware that, human cells have 46 linear chromosomes. And this is true for all eukaryotes, have multiple linears. Like yeast have 16 linear chromosomes, redwood tree has 66. And so we'll see this is common throughout eukaryotes. Another major difference is that our ancestral cells and our current cells have a remarkable ability to manipulate membranes. And when I say manipulate membranes, I mean we have the ability to use motor proteins and cytoskeletal proteins to push membrane out, as you can see here, to create additional surface area, or pull membrane in to create additional surface area again. This gives us all sorts of special powers in some ways. So we can do this repeatedly, and I'm going to show a few examples of this. So over and over again, we could pull in membrane. Now, let's add something new to it. Now, if you could, in addition to just pulling or pushing in membrane, you could pinch and fuse membrane. This is where things get a little bit special. By pinching and fusing the membrane, you can actually create an internal compartment. This isn't just additional surface area on the outside of the cell. This is actually a new room inside the cell. Now, bacteria don't do this that often. It does exist. It's just not nearly as extensive. And keep in mind that this internal compartment isn't going to be filled with cytosol. It's going to be filled with effectively extracellular fluid, the fluid from the outside world. And we can manipulate it, but it is a different substance. So we could continue to take in membrane, pull in membrane, and then pinch it. Pull in membrane, pinch it. Pull in membrane, pinch it, and create these internal compartments. Remember, these internal compartments are not filled with cytosol, but with effectively extracellular fluid. These spaces filled with non-cytosol are called luminal spaces. And the big advantage to these is that, one, you've got additional surface area facing outside fluid, so helps you with exchange of materials, wastes, and foods, etc. And then the other thing is that each of these compartments can be specialized for a specific function, meaning you can take all the machinery for one function, like digestion or toxin breakdown. And you can put all of the machinery to do that function in one room and really specialize all the componentry, increasing efficiency. So remember, this is going to be in opposition to cytosol, which is, the protein goo, the highly dense internal gel of our cells. Okay, now, if you haven't noticed yet, we've effectively created a new organelle, and I haven't even named it yet. Around our linear chromosomes, there is now a sort of double membraned moat. And this is now a nucleus. The nuclear envelope, again, double membraned. There are ways of getting in and out of the nucleus, but they're highly regulated. So random things like an invading virus doesn't just make it into our nucleus without any opposition. So this is the nucleus, basically supporting, surrounding, and maintaining our genetic material. Okay, in addition to the nuclear envelope, our membrane manipulations don't just stop there. There is an extensive set of elongations of this nuclear envelope. And These membrane extensions can pinch off into small packages, and these small packages can fuse together into new compartments. And each of these has a name. And I'll walk you through them. You may have even heard of these before. The first of these structures, the one that is directly continuous with the nuclear envelope, is the endoplasmic reticulum. There are multiple regions of ER, and that's the abbreviation. You may have heard of rough ER and smooth ER, and generally, smooth ER is for lipid synthesis, and that might be phospholipids or cholesterol or steroid hormones or something of that nature. And the rough ER is where we're going to package proteins for generally export. Now, these little round things, these are the packages typically filled with proteins set for export. These are called vesicles. And then the shipping center, where the vesicles will fuse in and then get repackaged over and over again, this is called the Golgi Apparatus or the Golgi bodies. Now, the Golgi is where the final destinations of each of these packages is determined. And so new packages are sent from the Golgi perhaps to the outside world, or material can be imported. Each of these processes has a name. The export process is called exocytosis, while the import process is called endocytosis. Typically, every cell is doing approximately equal amounts of these, though one might be more important than the other. They have to happen in approximately equal amounts so that one of these organelles we just talked about doesn't disappear from losing membrane. There is one more additional sort of major membrane manipulation event, which we'll call phagocytosis. And this is where you actually engulf something. This isn't just taking a little bit of fluid from the outside world or a few small molecules. It's actually engulfing something sizable, like an entire bacterial cell or even a smaller eukaryotic cell or debris. So here we go. Take the membrane, engulf an entire large structure. Now this structure, in this case a bacterial cell in orange, gets completely surrounded in membrane. And again, this process is called phagocytosis and is heavily requires cytoskeletal machinery, motor proteins, etc. Now, we're transitioning to the sort of last major topic for this video, which is probably the coolest topic in this video, which is the fact that approximately 1.8 billion years ago, give or take 100 or 200 million years, there was an independent heterotrophic bacterium, again drawn in orange here, which made its way into our cells, either because it got phagocytosed or maybe it infected our ancestral cells, and it took up shop. And was maintained and was able to reproduce. And if you haven't figured it out, this is actually one of our organelles now. This heterotrophic bacterium that was able to ingest food and or digest materials and generate ATP for its own purposes is now stably within eukaryotic cells, and we call them mitochondria. Now, this seems like a pretty crazy idea at first glance. We're literally taking two different, distinct parts of life's evolutionary tree and merging them into one. But there's a whole lot of evidence for this. Now, this was proposed by many people, but the person who deserves the most credit for really understanding the evidence behind it was Lynn Margulis of Boston University. And she laid out all of the different pieces of evidence that had been deciphered by other researchers and put them together into a coherent narrative. And so the first piece of evidence that she highlighted was, well, these mitochondria have double membranes. And that might indicate that it was a bacterium that got engulfed, where the outer membrane is the engulfing membrane and the inner membrane is the original bacterial membrane. So this is the first piece of evidence that you might look for. All right. So again, we're going to go through a few pieces of evidence that you would look for if this, that theory were supported by evidence. And so there's the first one. The second piece of evidence you'd expect if this were a ancient bacterium, is it should divide like bacterial cells. The way that bacterial cells divide is called binary fission. It is strictly a way of elongating a bacterial cell and after copying its chromosome, pinching in half. And this might be different from the version of cell division you may have learned in the past, which involves mitosis and cytokinesis, where multiple linear chromosomes are lined up and pulled apart. This is significantly different from that. And so bacterial cell division is exactly what mitochondria do. Okay, next. Another thing you'd expect to find in bacterium turned mitochondrion is you'd expect to have genetic material, and it does. Each mitochondrion has a single linear chromosome. So not only is there genetic material, it is the same size and shape as a bacterial chromosome, and sort of nail in the coffin of any naysayers, it actually has genes on it that are bacterial genes. It's not a complete bacterial set of genes, but the only genes on it are bacterial genes. There's a whole lot more evidence we don't have time to go into in detail. I'll quickly mention that these mitochondria have their own ribosomes, and they are bacterial ribosomes. There are multiple bacterial proteins in mitochondria, but we're going to leave it at that. Now, the next topic to extend this is to realize that after our proto-eukaryotic cell, effectively an archaeal cell, merged with a bacterial cell, we had a wide diversity of these that, diversified, to use the same term twice. One branch of that then did the same exact process with photosynthetic bacteria. Now, I'm not going to draw all of that in, but if a photosynthetic bacterium got phagocytosed and became stably associated within that cell, that's what we call a chloroplast. And there are multiple organelles that are all variants of this original photosynthetic bacterium, and we call these plastids, and we'll get to those another time. Okay, just a quick recap. Going back to the tree of life, we last finished up just looking at these four major branches of bacteria and archaea, where our ancestors, the proto-eukaryotes, are just effectively an archaeal cousin. Now, what we just talked about today is that our more recent ancestors are actually the merging of a proto-eukaryotic cell and a heterotrophic bacterium. And if we add on to that, the origin of photosynthetic eukaryotes is from an early eukaryote with mitochondria and a nucleus, also engulfing and incorporating photosynthetic bacteria, leading to chloroplasts. And this branch of eukaryotes is the branch that includes green algae and plants. Okay, hopefully that summarizes everything that you need to know for this particular topic. We'll cover more in future videos. Thanks, everybody. We'll talk to you again soon.